GeneCount DNA Purification Kit. This procedure is designed for fluids with low concentrations of suspended particles. Please contact LuminUltra if you would like to process a different sample type than what is recommended for this test kit. To get started, put on your safety glasses and gloves and set up your work area. Make sure that you have all the provided materials and equipment. This kit includes the following: a sterile 50 milliliter syringe, a syringe filter, the LT wash buffer A, a sterile 3 milliliter syringe, an extension tip, 5 milliliter tubes with LT lysis buffer A, a DNA cleanup column 1 with collection tube, a DNA elution tube, and 100 microliter filtered pipette tips. You will also need to gather several other pieces of equipment that are needed but not provided in each test kit, including one tube rack for 1.5 to 2 milliliter and 5 milliliter tubes, a microcentrifuge, and a fixed volume 20 microliter pipette and a fixed volume 100 microliter pipette. These items are available from LuminUltra as an equipment set should you need to purchase them. Refer to the chart below for the recommended volume for your sample type. Make sure to mix the sample well, then remove the plunger from a new 50 milliliter syringe and attach a new syringe filter. Be careful not to touch the tip of the syringe or the filter to avoid sample contamination. Pour the recommended volume of the sample into the syringe, then reinsert the plunger and slowly push the sample through the filter into a waste receptacle. Detach the filter and place on the tube rack so that it doesn't become contaminated. Then, remove the plunger from the syringe. Removing the filter before the plunger prevents damage to the filter from air being drawn back through it. Repeat the previous 2 steps if necessary until all the recommended volume of sample has been pushed through the filter. Make sure to record the total volume of the sample processed on the forms provided in your kit. If your sample has a high concentration of small particles and the recommended volume cannot be filtered efficiently, please record the total volume that was filtered and continue on with the protocol. However, please note that this may reduce the sensitivity of the procedure. Contact LuminUltra to determine if another protocol or filtration method may be a better option for your future testing. After all the required sample has been filtered, reattach the filter to the 50 milliliter syringe barrel and pour 20 milliliters of the LT wash buffer A into the syringe barrel. Reinsert the plunger and pass the wash buffer slowly through the filter and collect it into a waste receptacle. Push the remaining air in the syringe barrel through the filter to dry it. Transfer the syringe filter to a new 3 milliliter syringe and attach the provided extension tip to the outlet end of the syringe filter. Place the LT lysis buffer A tube in the tube rack and loosen the cap. Lower the extension tip connected to the syringe filter into the LT lysis buffer A tube. Slowly, pull as much lysis buffer as possible through the filter by gently pulling out the syringe plunger. If the lysis buffer shows evidence of precipitation before use, warm the tube in your hand until components are completely redissolved before using. Depress the plunger slowly to dispense the lysis buffer back into the same lysis buffer tube. Repeat the previous 2 steps 4 additional times. Be careful to pull and depress the plunger very slowly to avoid creating excess foam in the tube or the syringe. Slowly dispense all the lysis buffer back into the same lysis buffer tube. Gently mix the components of the new DNA cleanup column 1 and then shake contents to the bottom of the column before proceeding. Make sure to allow any cold columns to warm up to room temperature before use. Break off the tip of the column to remove and place the column into the provided 2 milliliter collection tube. Loosen the column cap a quarter turn and then centrifuge the column with the collection tube for 1 minute at 3000 RPM to remove the storage buffer. Always be sure to balance the centrifuge before starting. Refer to the troubleshooting guide and the written instructions for more details on how to do this. Place the column in a new, empty 1.5 milliliter elution tube, and then tap the column in the elution tube firmly on the bench 3 times to ensure that the resin is completely compacted. Slowly apply 100 microliters of the lysed sample to the top of the compacted resin bed, taking care not to touch or disturb the resin. Loosen the column cap a quarter turn and centrifuge the column with the elution tube for 2 minutes at 3000 RPM. Once again, making sure to balance the centrifuge before starting. Once the centrifuge is complete, remove your elution tube and label it. The purified DNA is now in the elution tube and is ready for immediate qPCR analysis. Discard the column after use. You are now ready to transfer your DNA to the assay tubes so that they can be tested in your qPCR device.